going to pick up with our discussion of systematics, looking at prokaryotic classification. And one of the difficulties in classifying prokaryotes, there are a number, but one is that they're just very, very small. 2 micrometers is typical size of a prokaryote. So we rely heavily on genetic analysis. We look for genetic signatures to define groups of prokaryotes, and there are actually some problems with that, but again, it is one of the main tools that we use in prokaryotic classification. And then we also rely very heavily on biochemistry as well. Now, again, we've talked about some of this before, but cell wall composition is very important, or a first step, for example, in looking at characteristics of prokaryotes. And again, biochemical or physiological pathways, heterotroph, autotroph, oxygen tolerance. Also things like endotoxins and exotoxins. An exotoxin is actually something that's toxic, and it's produced by the bacterium and released outside the bacterium, gets into our bloodstream and so on, potentially causing problems. An endotoxin is actually part of the physical makeup, typically the cell wall or the capsule of a prokaryote. So endotoxins are part of their normal structure. And when those organisms die, then the capsule or the cell wall is toxic as well. Also limiting nutrients when we try to grow bacteria. As I mentioned in the previous module, when we use agar plates, bacteria can't digest the agar, but we can put specific nutrients in the agar, and depending on what nutrients are in the agar, that will determine what bacteria or archaeans can grow. And then also another characteristic that can help us distinguish is that some bacteria and other prokaryotes are nitrogen fixing. And in nitrogen fixation, what happens is that atmospheric nitrogen, N2, is converted into ammonia. And atmospheric nitrogen really doesn't have many biological uses, but ammonia, NH3, does. I think I may have said NH2, but ammonia is NH3, and so that does have biological uses. Plants and other organisms can absorb that and then convert that and utilize that in amino acid metabolism, for example. So anyway, so that's an important property. And so again, we'll talk about some of the organisms that do this, but one would be what we call blue-green algae. They have specialized cells that actually fix nitrogen from the atmosphere and then convert it to a usable form for everybody else. And there are other biochemical pathways as well, but these are just a few that we typically look at. Now also, as I talked about last time, colony characteristics and also the behavior of bacteria can also help us in classification. So we normally think of bacteria and archaeans, the prokaryotic organisms, as being unicellular and not having anything to do with one another, but they can form associations to form what are known biofilms. Basically, the bacteria secrete products that kind of glue them together, and they can actually communicate with one another. And then also they sometimes have behaviors for producing spores, protective capsules around the cells and so on. So again, the behavior of individual cells can be important. And then also, as we talked about last time, and I'll just kind of quickly show you this, is depending what kind of colonies they produce on augars. So for example, we looked at this before, but some prokaryotes can be different pigments, and also the margin of the colonies may be different. So some may be smooth and kind of mucus looking, others are feathery looking. But there are a number of properties we can look at in terms of colony morphology that will tell us what type of organism we're dealing with. So sometimes we don't even look at the bacteria, we just look at their biochemical characteristics and we can classify them accordingly, or just look at their genetic makeup, quite honestly. Okay, morphology is utilized in classifying prokaryotes, but it's just not as useful because they are so tiny. But terms that we use to describe the morphology of prokaryotes, one is coccus or cocci would be the plural here, and that would be a spherical bacterium. And so spherical bacteria are going to be cocci, and streptococci are those that form chains of spherical bacteria. Staphylococcus or staphylococci would be those that form clusters of spherical bacteria. And diplococci are those that generally form pairs of spherical bacteria. So again, these would be some examples of different morphologies of bacteria. Additionally, the term bacillus refers to any rod-shaped bacterium, or bacilli would be plural. And so again, basically rectangular looking organisms. And you can see here why this is actually at 1,000x, multiplied or magnified 1,000 times. These are rod-shaped bacteria, and you can see how tiny they are. And again, for the untrained eye, a lot of times rod-shaped and spherical and so on all begin to look alike. So again, it does take some time and really good microscopes to be able to distinguish morphologies. Having said that, I'll continue to talk about a few other morphologies. One would be spiral-shaped bacteria, known as spirillum or spirilla. And the spirilla will have external flagellae. So that is one thing. If they do have flagellae, they'll be external flagellae, because there's another type of spiral-shaped bacteria known as spirochetes. And the spirocheta are interesting because their bacteria actually are embedded in the cell wall and go back over the organism itself. And so the spirochetes, as they move then, as the bacterium tries to spin, it basically causes an undulation, kind of a corkscrew, as it moves over a surface movement of the animal, or excuse me, of the prokaryote. And some spirochetes are responsible for a lot of pathogens. Gonorrhea is one, and then also Lyme disease is another. So Lyme disease is carried by ticks on the East Coast and produces a characteristic rash, but that's a disease caused by spirochetes. And then also Vibrio bacteria are those that have basically a comma shape. So it's like a bacillus, but if you bend the bacillus a little bit, that would be Vibrio. All right, so again, morphology has limited benefits, but we do use it as part of characterizing prokaryotes. And there are other features we might consider as well, but those are the most typical. Now, when you look at prokaryotes, there are basically two ways that they reproduce. One way that they reproduce, and that would be an asexual reproduction known as binary fission. And in binary fission, you may recall that bacteria have a circular chromosome, and so that chromosome is replicated, and then growth will occur in the cell wall and cell membrane between the attachment sites of these two chromosomes, and the cells will elongate, new cell wall built in, and we end up with two cells. So they mainly reproduce through binary fission. However, they do have a sexual process, which is known as conjugation. And so it requires a special kind of plasmid. And you may recall that in addition to chromosomes, prokaryotes do have smaller loops known as plasmids. And so there's a special kind of plasmid which triggers a reproductive cycle. And so it will cause production of a conjugation tube between two different types of prokaryotes. And then recall, of course, the DNA is double-stranded. And so one of the strands of the plasmid then will peel off. Basically, it breaks and peels off, moves through the conjugation tube, and then reforms in the other bacterium or archaean, in the other prokaryote. And then the complementary strands are rebuilt in each of the prokaryotes. So at the end of the process, then you have both bacteria with that particular plasmid. Excuse me. Now, what can also happen is that the plasmid can be spliced into the chromosome, and actually portions of the chromosome can also be transmitted through the conjugation tube. So it's not really a sexual reproduction then, but it's a sexual process in a way that a transfer or exchange of genetic material can occur. And this is one of the ways, for example, that bacteria then transmit resistances to antibiotics. And in the past, we thought that these prokaryotes had to be closely related to conjugate with one another, but we now realize that really very unrelated types of bacteria can conjugate. And so this is one of the places where, common genes don't necessarily tell us about the vertical flow, from parent to offspring kind of generation. There's what we also have is a horizontal, what we call horizontal flow of genes between unrelated organisms, or between horizontal flow between unrelated organisms of genetic material. So even archaeans and bacterians can actually share genetic information. So anyway, this is the problem with antibiotic resistance is once they develop, they get passed within the microbial community pretty quickly, and it's one of the real challenges we face in terms of health in the coming century. Okay, now, as we look at the domains then, domain eubacteria, we talked about previously before, but what I want you to remember, among other characteristics, is that they have unique cell walls. So they are prokaryotic. They have what we call polycistronic mRNAs, meaning that their mRNAs code for multiple proteins. But the key thing there that makes them unique is unique cell walls with peptidoglycans. The cell wall, when we look at the bacteria, there's two general categories of bacteria, gram-positive bacteria that have a thick layer of peptidoglycans outside of the cell membrane. And so this will hold a pigment known as crystal violet. So they normally stain kind of a purplish color. And then gram-negative bacteria, which have a thin layer of peptidoglycans, which is actually sandwiched between the plasma membrane and another membrane external to that. And so they do not hold crystal violet very well and tend to stain kind of a pinkish color. So when we look at a mixture here, the purple bacteria here are going to be gram-positive, the pinkish ones are gram-negative. Now, examples and types of eubacterians, again, there are many, many types. This is just kind of the tip of the iceberg here, but one group would be what we call endospore-forming bacteria. Now, the term spore I'll be using a lot. A spore basically is a cell that has a heavy cell wall around it. They can typically tolerate desiccation, dehydration. Sometimes they can tolerate temperature extremes or changes in acidity. So it's basically a way for an organism to get through difficult times. Anyway, the endospore-forming bacteria form spores kind of at one end of the cell, and again, is kind of notorious for a number of pathogens. The genus that causes tetanus, botulism, would be in this group. The genus Clostridium would be one example. Enteric bacteria, those that we find in our intestinal tract. E. coli would be an example here, Salmonella and others. Many of them can be pathogenic, causing severe diarrhea. Spirochetes would be another major group of eubacteria, and of course the spirochetes have their characteristic flagella embedded in the cell wall. We talked about Lyme's disease and gonorrhea being a couple of diseases caused by that. Gram-positive bacteria would have gram-positive cell walls, the thick peptidoglycans layer, and again a very diverse group filling a lot of different niches in nature. Some are pathogens, but many of them we find in our soil doing all kinds of important recycling activities. And then another really important group are the cyanobacteria. Their common name are the blue-green algae. And the cyanobacteria practice plant-like photosynthesis, have the same basic pigments as photosynthetic chloroplast, and this is the group that is ancestral to the plant chloroplast. Now, the domain archaea. Second major domain, again, what I want you to remember, of course, certainly is that they're prokaryotic, but really what distinguishes them from the eubacterians is that they have the unique plasma membranes, branching phospholipids that go all the way across the membrane. So that's what's unique about the archaea, in addition to being prokaryotic. And here examples or groups within the domain archaea include the extreme halophiles, which means that they can tolerate extreme salinity, a lot of high concentration of salt. The hyperthermophiles, which can tolerate extreme temperatures. So in the hot springs, we typically find a lot of archaeans. And also, when you go to hot springs, the different colorations that you see there are typically caused by archaeans and also heat-tolerant bacteria, and even some heat-tolerant eukaryotes cause the colorations that we see. And then also methanogens. These are what produce, quote-unquote, natural gas. So when you have to pass gas, methanogens are responsible for that. They're feeding on what you don't eat in your intestine and then produce methane gas. And there are a number of other archaean domains as well that we have recently discovered. So the archaeans are generally considered quote-unquote extremophiles, that they like extreme environments. But what we are learning and discovering over time is that really archaeans are found in almost all environments. And even eubacterians are found in quote-unquote extreme environments. So anyway, we're learning more and more about archaeans all the time. Okay, that then brings us to the domain eukarya. And of course, the eukarya are distinguished by being eukaryotic, and eukaryotic cells are typically large, much larger than 10 micrometers typically. Now, there are some terms that I'll be using as we talk about the eukarya. And these are not really taxonomic terms, they're more descriptive terms. So for example, I could describe us taxonomically as Homo sapiens. That's a scientific term, scientific name for us, composed of the genus and the species name. But I could also call myself a human being. That would just be a descriptive term. That really doesn't have a scientific designation to it per se. So when we talk about eukaryotes, for example, the term Protista is one that's used a lot, and that's a reference to any unicellular eukaryote, or is, for example, an amoeba. It could also refer to what we call colonial eukaryotes, and those are eukaryotes that loosely associate and can communicate, but they aren't obligated to be in that multicellular configuration. It's an optional configuration for them because they can live individually if they need to. And then also multicellular eukaryotes that lack tissues. And a tissue then would be specialized cells, groups of cells that are specialized for some function. So different types of algae, like our brown kelp would be an example of this. Now, other terms within the Protista, protozoa refer to the so-called animal-like protozoa, and by that I mean heterotrophic protists. Ciliates would be any protist that is ciliated. Uses cilia for locomotion. So cilia, you may recall, are hair-like structures on the surface of cells that they use for swimming. Flagellates would be any protist that use flagellae for moving from place to place. And so again, flagellae beat back and forth, pull them through water or body fluids. Amoebas are organisms then that move via structures known as pseudopods. And in pseudopods, and actually I'll go back to this. So when you talk about, I'll talk about amoebas later, the movement of amoebas later, but basically they can change shape and change form. So the pseudopod constantly transforms shape, but I'll get back to that in a few minutes. And then again, the term algae is a reference to the multicellular protists that lack tissue differentiation. And they're also photosynthetic. So the algae can be unicellular as well. So they can be the unicellular protists that are photosynthetic, or multicellular that are photosynthetic, or colonial forms that are photosynthetic. Now, within the eukarya then, we recognize 4 superkingdoms. The Excavata, and I'll come back and describe these in more detail in a few minutes. A group known as the SAR, and you don't necessarily need to know what that stands for, but it stands for subgroups Strumenopila, Alveolata, and Rhizaria. The Plantae, and the Uniconta. So I'll be discussing these 4 superkingdoms and examples of each. So starting with the superkingdom Excavata then. What distinguishes them is that they lack classical mitochondria, the kind of mitochondria that we find in really all the rest of the eukaryotes. And many of these are pathogens because they are not very efficient at generating energy, so a lot of times they're intracellular pathogens of other organisms. Some examples of Excavata include the diplomonads, and an example of that would be a Giardia. Giardia causes a very severe type of diarrhea. So the Giardia organism is a small flagellated organism, really not much bigger than bacteria. They don't produce a lot of ATP, so they're very inefficient, so they are obligate parasites. And so anyway, Giardia is found throughout the western United States for sure, really all over the world, and causes a very severe form of diarrhea. And so you really have to be careful if you're out hiking and so on. You just can't drink water from anywhere. You need treated water. We need to be careful about that. Trypanosomes would be another major group. They're a flagellated parasite. They also have what we call an undulating membrane. So they actually have a flagellum that's embedded in the cell membrane, and it forms a sheet-like structure. And these are blood-borne pathogens, and they cause 2 major diseases: Chagas disease in Central and South America, that's transmitted from person to person by a kissing bug, and then also sleeping sickness in Africa is caused by a trypanosome, and that's transmitted by the tsetse fly. And then the hypermastigophora would be a third example of or third group within the Excavata. And the hypermastigophora, what the word is trying to tell us is that they have lots of flagellae. A mastigophora is a reference to flagellae. So this is an electron micrograph, and you can see how many flagellae these have. And it's the hypermastigophorids that live in the guts of animals that are capable of digesting cellulose. So hypermastigophorids live in the guts of termites, goats, deer, other herbivores, and they digest cellulose and produce sugars so that the animals can survive, basically. So it's a mutualistic relationship with the host animal. Okay, our next superkingdom SAR. Some have a tetramembranous chloroplast. So what that means is instead of the chloroplast having 2 membranes, it will have 4 plasma membranes. So that's unusual. They exhibit either flagellae or cilia. Some of them have what we call hairy flagellae, and usually in combination with the smooth flagellum. So here's this hairy flagellum here, smooth flagellum here. So that's particular to this group. Some have invaginations of the cell membrane, and then some locomote via pseudopods, the amoebas. So looking at examples of SAR, subgroups within the SAR, one subgroup would be the rhizopods, or the amoebas. And amoebas, as I've talked about, move and feed via pseudopods. And so the pseudopods can be thread-like and flexible, they can be thread-like and stiff, or they can be more lobe-like. And so I want to quickly show you, what it looks like when an organism is able to move via pseudopods. And so this is an example of an amoeba here. And so again, these are the pseudopods. And you can see that the pseudopods basically change shape constantly. And so what the animal can do is actually change from a fluid state of cytoplasm to a more gelatinous state of cytoplasm. And so it's known as cytosol and cytogel. And as it makes these transitions, it's much like what happens when our muscle is soft and our muscle is rigid. So anyway, I think that shows you then that the shape of an amoeba then is ever changing. And that's one of the things that's interesting about the amoebas. Okay, another group of the SAR are the apicomplexans. And so the apicomplexans have at their tip a complex that helps them get inside of cells. So all of the apicomplexans are intracellular parasites, and they cause a number of different diseases. Probably the most famous disease that's caused by an apicomplexan is malaria. So malaria is an apicomplexan disease transmitted from person to person via the mosquito. Another subgroup of the SAR are the ciliates. So the ciliates are covered with cilia and swim via cilia. They also have two types of nucleus, a macronucleus and a micronucleus. The macronucleus is polyploid, carries out the day-to-day activities of the cell. Micronucleus is involved in a sexual reproduction for them, or sexual process, I should say. Euglenoids would be a small unicellular algae. It's also flagellated, and they're found in a number of different ecosystems, mostly freshwater, but all over the world. Dinoflagellates would be another unicellular algae, so these are photosynthetic organisms. And the word here is telling us that they're spinning flagellates. So the dinoflagellates, again, are found mostly in marine ecosystems. They have two flagellae, a horizontal and transverse flagellae, and so as they swim, they spin. And that's where the term comes from, dinoflagellate. The dinoflagellates are also interesting because they're responsible for the red tides that we have along our coastlines, and many of them are also bioluminescent. So if you've ever seen bioluminescence in the oceans, it's primarily due to these guys. And if you've never done it, I mean, if you've ever gone to the beach when there's a really intense red tide, no moon, and big waves, it's just spectacular because you'll see incredible bioluminescence. Diatoms are another unicellular algae, found in a number of marine and aquatic ecosystems. They are noted for having two shells that kind of fit together like a petri dish. You can see that there's some flagellae here, but again, a major part of the unicellular ecosystems. And all of these unicellular algae collectively do produce a significant part of our atmospheric oxygen. So as we look at the health of the oceans and aquatic ecosystems, we have to take care of those because, again, that's where a lot of our oxygen comes from. Diatoms are also really interesting because there's tremendous diversity. They have a shell that's composed of silicon compounds. They're glass-like shells, and just extraordinary diversity among the diatoms. The golden algae are relatives of the diatoms. Can be unicellular or form colonial organisms. Again, aquatic, some marine examples as well. Also contribute significantly to atmospheric oxygen. And then the Oomycota are a fungal-like group. And I mention them because they are significant because they are pathogens, for example, of plants. They're significant pathogens of plants. The potato blight that originally afflicted Ireland, for example, is caused by the Oomycota. And so, again, they form thread-like structures that are called hyphae. The mass of thread-like structures is known as a mycelium. But these mycelia, again, some of them are what we've considered detritivores, or basically feeding on dead organisms. But they also are pathogens of both animals and plants. Another example would be here in California, we have something called the oak blight. The oak blight is also caused by an Oomycotin. So it's not a true fungus, but it's a fungal relative. Slime molds also are at one time were classified with fungi, but have been separated out. And the slime molds are kind of interesting because, again, there are two variations here, some that are cellular and some that are acellular. And so I'd like to actually show you the acellular slime mold and how they move here real quickly. And so these acellular slime molds do not have individual cells. They form what we call a plasmodium, which is just kind of like a shared cytoplasm. And they stream across surfaces. And so you can't see it very well, but the cytoplasm actually streams much like an amoeba's cytoplasm does. And so they crawl along the surface of forests and so on, decomposing vegetation as they go. They're usually brightly colored, so they're really kind of spectacular looking organisms. But anyway, so they also fit into this group known as the SAR, and then they will form specialized structures for reproduction. And then still another group are the choanomastigophora. And what this is trying to tell us is that they are flagellated, they bear flagellae, and they're what we call collar cells. They have a kind of a collar-like structure associated with them. And so this is actually from a sponge, but it's a similar type of cell. So this is the choanomastigophora in here. So anyway, the importance of the choanomastigophora is that that is the group that we consider to be ancestral to the animals. Now, another superkingdom are the Uniconta. And what distinguishes the Uniconta is that they have a single flagellum, they are heterotrophic, and most members are multicellular. Now, examples of the Uniconta then are both fungi and animals. And so in terms of different types of fungi, and we'll come back to these in our next module, but examples would include things like mushrooms, morels, if you're familiar with those. But also, if we can go to the Elmo here real quickly, also just molds, like black bread mold would be an example of a fungus as well. And then, of course, another member of the group then would be animals. So, and another term for the animals are the metazoa. So, we won't really have time to get into our last superkingdom, which will be the superkingdom Plantae, which we'll also continue in our next module. So I'm going to come back to in our next module talking more about fungi and their characteristics, and then we'll also consider the Plantae as well. And then as we get into the Plantae, what we'll learn is that they all have kind of a unique life cycle where they alternate between a diploid generation and a haploid generation. So one of the things I want to foreshadow a little bit is that in our next module, as we talk about plants, we'll want to talk about this alternation of generations, how they go from a diploid generation to a haploid generation. So, as we look at the eukaryotes then, tremendous diversity, 4 supergroups, and we'll talk more about those in our next module.